On behalf of the organizing committee of the 33rd Argentinian meeting of plant physiology, I welcome you to the satellite workshop. Will the Nagoya Protocol change how we deal with digital sequence information in plant research and law perspectives? We would like to thank the Global Plant Council for organizing this workshop. We have three prestigious speakers today: Dr. Amber Hartman-Scholz, Dr. Shane Anderson, and Casey Bansal. My name is Fernanda Gonzalez, and together with Isabel Mendoza, we will be coordinating the workshop. You can write your questions to each speaker in the question and answer chat. At the end of the three talks, we are going to read and answer them. And you can also vote which question do you prefer. And we will start with the one with more likes. And you can also raise your hand if you want to ask live. Well, our first speaker is Dr. Amber Hartman-Scholz. She's the deputy to the director of the Leibniz Institute, DSMZ, the German Collection for Microorganisms and Cell Cultures in Braunschweig, Germany. She headed the team that led the DSMZ, becoming the first registered collection under the Nagoya Protocol in the European Union. She was also the head author of the Combined Study on Digital Sequence Information in Public and Private Databases and Traceability, submitted in January 2020 to the parties to the Convention on Biological Diversity. Amber is currently leading a German-funded project called Wild DSI on DSI and Open Access, which recently released a white paper on DSI policy options. She is deeply engaged in access and benefit sharing, DSI, CBD, and Nagoya Protocol issues. Dr. Scholz has broad experience in science and policy. She was executive director to the President's Council of Advisors on Science and Technology in the White House and science fellows to the Environmental Quality Committee within the California State Senate, USA. She received her PhD in biology from the Johns Hopkins University with a focus on the human intestinal microbiome, microbial genomics, and bioinformatics in 2009. The title of her talk is Nagoya and DSI. What we are talking about? Well, dear Amber, whenever you want, you can start your presentation and share your screen. We are going to close our cameras and microphone.
Perfect. And can you see my screen, my slides okay? Yeah?
Yes,
thank you
perfectly.
Perfect. Okay. Thank you for the kind introduction, Fernanda. And thank you for inviting me to join you all the way over there in Argentina this morning. I would love to be there in person. I'd like to kind of give you a high-level overview of first what the Nagoya Protocol kind of is, but I'm not going to spend a lot of time there. I'll jump pretty quickly into the current political debate around sequence data and the potential ramifications that that might have for scientists, and also the way that scientists can be involved and contribute to the science policymaking process. So hopefully, even though you're plant people, probably most of you, you've heard at least of the Nagoya Protocol. So the Nagoya Protocol came into force in 2014. It was negotiated in 2010, and it is basically a supplemental agreement to the 1992 Convention on Biological Diversity under the United Nations Environment Program. So the full name of the Nagoya Protocol is there in small letters. As you can see, it's long. It doesn't fit very easily on a single big slide. The Nagoya Protocol on Access to Genetic Resources and the Fair and Equitable Sharing of Benefits arising from their utilization. And as a protocol to the CBD, as I mentioned. And to make a long story short, there basically are two main purposes of the Nagoya Protocol. First, to provide more legal certainty for the actors that are involved in the use and exchange of genetic resources, as well as traditional knowledge, and to specify, to clarify exactly how the sharing of benefits should happen. Now, to understand why somebody would go about having the Nagoya Protocol, you have to kind of look back to the 2 decades before then, when the Convention on Biological Diversity established the idea of access and benefit sharing but yet the procedures for how this should be done were relatively unclear, or they were not legally binding. And so the Nagoya Protocol takes that idea and codifies it in a more defined way. What is really important for researchers, for life scientists to know is that it is, as probably might be suspected by talking about genetic resources, it includes DNA and RNA, but it also includes the whole organism. So plants, animal, fungi, microbes, and as well as their biochemical compounds, also sometimes known as derivatives. Importantly, and a mistake that many researchers tend to make when they're first entering the field, is that it applies both to commercial and to non-commercial research. There is no carve out that says, oh, you're doing basic research, you're out of scope. All use of genetic resources is under here. Now, there are some important exceptions, but I think it's probably better if we don't concentrate on the exceptions, but on the general principles for now. To break it down a bit further, we put together this slide through the Nagoya Hub project that I lead over here at the DSMZ for German researchers. In a nutshell, ABS, access and benefit sharing, is about first access, taking and using material for research and development, and asking the providing country's permission first. Benefit sharing means giving something back. That can be, in the case of a commercial project, certainly monetary benefits, but in the case of non-commercial benefits, it could be things like co-authorships or scientific training, or perhaps technology transfer. That means that there's a wide spectrum of activities that are envisioned under the Nagoya Protocol beyond just monetary benefit sharing. And in theory, it would also be ideal if these types of benefits contributed to conservation and sustainable use. Now, although the Nagoya Protocol codified benefit sharing, each country still is able to do its own thing, to choose how it wants to do it. And because of that autonomy that each country has, what was envisioned is a central clearinghouse, seen here in the ABS clearinghouse, which means if you know that you're an ABS case because your country has something to do with CBD or Nagoya, then in order to figure out how you're supposed to navigate the system, the primary contact point is supposed to be the ABS clearinghouse. And then you navigate the rest of the system and the documents, the permits that are needed, and usually report back to the authorities. The documents that are foreseen under the Nagoya Protocol have alphabet soup names. PIC is prior informed consent, MAT is mutually agreed terms, and then many scientists and their respective universities are very familiar with MTAs, material transfer agreements. Now, unfortunately, MAT and MTA are similar, but actually envisioned as, in theory, different documents. The other important aspect that many parties have unfortunately overlooked is that the Nagoya Protocol did actually create an economic marketplace, if you will, where for those researchers that have the choice over jurisdiction in which area they want to work, if the country does not require ABS permits, the path to compliance for that user actually becomes very simple. And certainly what we've seen in my field, in microbiology, where we are dealing with many organisms that are cosmopolitan, which means they can be found in many different locations, you start to see that countries that have very heavy ABS processes start to actually lose out, because if the process gets too complicated, it can be very difficult for them to justify working in that country. So here I took a screenshot from the ABS Clearinghouse website for Argentina. You can just type in ABS Clearinghouse into Google and then type in Argentina, and you'll find out what the rules are. The other problem, of course, that researchers have is sometimes when there's not sufficient information about a country available. And Argentina might be sort of a mixed example here. You can see down at the bottom, you do have information on your national focal point, on your competent national authority, but there is no information on the legislative or policy procedures or ABS procedures. You can see there's a 0 there. And so imagine if you are a researcher that from Germany that wants to go work in Argentina, or an Argentinian researcher, and you want to use your resources and send them outside of Argentina, you would have to know, well, how do I comply with the legislation, with the procedures that are in place? Well, you'd have to email the person, ask them, hope they respond, but yet you're not able to sort of self-inform yourself about your obligations ahead of time. And this is actually, unfortunately, quite common in the ABS, the world at this time point. I'm speaking to plant physiologists, and so I think it's really important to draw attention to this excellent review by Sylvain Aubry. illustrating that it's not only, of course, the Nagoya Protocol that you all need to worry about. You also have the international treaty, and I think Casey later on today will tell you about that, but you have an even more complicated situation where you're dealing with the Nagoya Protocol if the plant species that you're working on is not listed in Annex 1 or is not under the multilateral system, or if it's in the MLS and or it's in Annex 1, but you're researching not for food and agricultural purposes. So then you slide down into CBD space. Now, if what I said made zero sense, that's okay. Maybe that's also illustrative of the fact that it can be very difficult to figure out your legal situation, especially if you're relatively new to the field. It's important to know, first of all, there are multiple different legal obligations that plant research have, different mechanisms that can apply to you under different conditions, and you need to seek out expertise from your university, from your collaborators, if you don't have a way of figuring out this yourself. I know that's a little bit of a mystery, and this will actually become important as we now discuss DSI. So what was supposed to happen in May of 2020 was the Conference of the Parties to the CBD, COP15. This, because of the pandemic, this UN convention has been postponed now until May 2022, so 2 years later. But no matter what, political experts of all stripes and colors believe that at COP15, whenever that actually takes place, a political deal will be sought. And that will be a deal or a trade-off between a big UN biodiversity framework called the Global Biodiversity Framework. It was called the post-2020 Global Biodiversity Framework, but now it's 2 years post-2020, and so it's now just being called the GBF. And really a trade-off with digital sequence information, where there is a bit of a horse trade, a political type of log rolling that's sought between these two. The reason for that is because the previous strategic plan for biodiversity envisioned in some way in the Aichi targets did not work. So you can see this small infographic here, tiny bullet points, but all of these are different measures of progress on biodiversity loss. And you can simply see the number of red arrows here. And that means we haven't done enough to protect biodiversity for conservation. And so the new post-2020 GBF will actually set regional goals, have quantitative indicators, and be more specific. Now, in order to get parties to agree to that, the developed world will need to trade something in exchange to find consensus among all parties. And what's likely to happen is that there will be, and there already is, very high pressure to figure out what to do with sequence data that in theory could be under the Nagoya Protocol, but in practice is not really being handled that way. What's important is to kind of look at the data. So you can see here, this is on the x-axis time, on the y-axis, the cumulative equivalent of a number of human genomes. So basically a proxy for total volume of sequence data in the databases. And back in 2010, when the Nagoya Protocol was negotiated hand in hand with the Aichi targets that I just showed you, the databases were full at the time, but the exponential increase in data was huge over time, right? And so what parties have said as they've continued to meet every 2 years since the Nagoya Protocol was negotiated, hey, look, these sequence databases are growing. They're a lot bigger than we ever realized they were going to be, and they're all open access, which means researchers have an endless amount of data they can access. And on top of that, they have these new tools for synthetic biology, right? In 2020, the Nobel Prize in Medicine for CRISPR-Cas9, the so-called genetic scissors, incredible precision in editing molecular sequences. And looking over here, synthetic biology has given us all of these tools listed in this table here on the left. And you can see by looking at the number of stars in each column, you can see, yeah, look, the technology is not perfect, but in a lot of places, there are a tremendous amount of techniques that have been developed in the last decade that enable us to go to the database in silico, to apply the synthetic biology tools, and to not need to go back to the country of origin, to go back to Argentina and ask for permission from the competent national authority. Can I use this? So basically, parties are arguing there is a loophole in the Nagoya Protocol, and that is, the bad guy is the open access databases where sequence data is stored. I'm putting this, simply, but that is fundamentally the argument that's being made. And what you see in the political space is that the way that sequence data, which is called digital sequence information, so we'll use the acronym DSI, is being negotiated in this intersessional period between COP14 and COP15, is that it is directly tied to something called the Open-Ended Working Group 3. The Open-Ended Working Group has the explicit task of building up the global biodiversity framework. And actually all the deliverables of the intersessional period do not feed into something called the SubStuff, which is the subsidiary body on scientific, technological, and technical advice, as they normally would, but they feed into another body negotiating the GBF. This indicates to us that DSI is a make or break issue, meaning that parties have basically forecast that DSI is a must negotiate issue if we want to have the global biodiversity framework successfully negotiated. You also see the political tensions that arise when you look at the number of studies, the number of different areas that parties are discussing. And as Fernanda so kindly mentioned in my introduction, I was the lead author on a combined study. It was supposed to be 2 different studies on databases and traceability for sequence data for DSI. And actually from that study, I'd like to give you a few take-home messages of what we learned there. So first of all, we noted that although there is tremendous progress in synthetic biology and that one might like to apply a Nagoya Protocol-like system to sequence data, that probably won't work because the data ecosystem for sequence data is not like the Nagoya Protocol. What do we mean? Well, first of all, there are 212 billion sequences in the databases from every country in the world. The entire DSI data set, all 212 of those, are downloaded 34 million times a year by different users. There are about 10 to 15 million users also in every country in the world. And then this data is also electronically and automatically shared with 2,000 downstream databases. All of this data is then cited and referred to, referenced to, in about 500,000 publications and patent applications. So it's a big data world, very complicated, highly interconnected, and a high volume. It is not well suited to sort of the paper stamp type of system that the Nagoya Protocol envisions. Furthermore, there are a number of myths that exist in the broader CBD context where access from biodiversity-rich countries, such as countries near the Amazon or in Southeast Asia, provide access to their biodiversity, and it's mainly used by researchers in developed economies. In fact, over half of the DSI in the database was sourced from high-income countries, and most of it does not come from, at this point at least, low and middle-income countries. So you can see China, United States, Canada, and Japan. These are also countries that do not require ABS access permission. Now, this becomes really important when you think about creating economic incentives for wanting to have biodiversity-rich countries be able to profit from their biological diversity, be it in the physical resource itself or from their data. Furthermore, there are 10 to 15 million users of this database, and they live in every country in the world. Those user countries tend to be, again, similar to the top players that we saw before. And in Argentina, I looked at the numbers, there are about 30,000 plus Argentinian researchers that use GenBank on a regular basis. Importantly, these infrastructure costs for maintaining this core data infrastructure, which is called the INSDC, the International Nucleotide Sequence Database Collaboration, which is 3 databases, GenBank, ENA, the European Nucleotide Archive, and DDBJ in Japan, which are identical databases that share data. Those costs to maintain that database cost about $50 million per year, which is about $3 to $5 per user. And about half of those users actually live in countries that don't contribute to these costs. Now, interestingly, Argentina is actually a member state to EMBL, to the European infrastructure that supports the European database. So actually, Argentinian users are one of those countries that do contribute to infrastructure costs, or that the government pays a certain membership to maintain that. But many countries around the world do not, and you might consider those benefits really to be truly a type of non-monetary benefit provided by these open access databases. Now I'd like to take you kind of through a tour of ways or perspectives from scientists that can be used and are being used in the policy-making process around DSI. So I led a project called Wild DSI, which is a German acronym that talks about DSI. And really the starting point of this was the recognition that the GBF, the Global Biodiversity Framework, would require a compromise of some sort on sequence data and benefit sharing. And we really asked the question, how can the scientific community contribute to this policymaking process and provide new data? The first thing that we did was to conduct a variety of stakeholder workshops, and we have lots of little examples of how open access to DSI, to these 212 million sequences, contribute to conservation, to sustainable use, to global health, to the public good. And here's just a couple of screenshots from the different presentations that we heard during these stakeholder workshops. And you can see there's a wide range of different public good that takes place with DSI. But of course, the fundamental argument is that also commercialization can take place with these sequences. And that's where the parties that are negotiating on DSI would like to figure out how they can harness some benefits from this data. We put together a white paper back in October, almost a year ago now, called Finding Compromise on ABS and DSI under the CBD, and putting forth 5 options for open access options that can still contribute to benefit sharing. We outlined the requirements that would be needed for scientists to be able to work in a new DSI mechanism. We spent a bit more text than I have here, but it's a list of 5 things. We call for open access. We call for a simple system that it probably shouldn't look like the Nagoya Protocol per se, because the data ecosystem, the big data world, doesn't match that model. We also talk about the idea of future-proofing a DSI mechanism, meaning that the data itself doubles in size every 1.5 years, and it continues to expand into new types of omics fields, from metabolomics to proteomics to microbiomics and so on. And it needs to be aware of future technological innovations so that we don't have to keep reinventing the wheel every 10 years in the policy space. It also needs to create legal certainty. So I showed you that the green diagram earlier of the different legal frameworks that, for example, plant scientists must follow, and creating yet another system in even more complexity does not create legal certainty. And finally, we also point out that for those scientists that physically access material in country and plan to sequence it, that it might be nice if the physical access systems were compatible with the sequence, the DSI mechanism. So an opt-in potentially for genetic resources. We also note that the sequence databases are complicated. They contain everything from all fields of life sciences, from teeny tiny viruses or plasmids, all the way up to huge, massive plant genomes. And they fall in a legal way under many different legal purviews, under different UN constructs. And what would be really sad and perverse is if all of these different UN bodies started to make DSI rules on benefit sharing that were not compatible with each other. Because then if you did a blast search for your favorite gene, then you might actually be under 4 or 5 different UN frameworks ultimately, right? And I think that the idea of, or the concept of scientists really explaining how they use the database, how they use the data sets, and explaining to policymakers why we would have a problem and that we don't know what we're looking for until we're in the middle of a search is an important concept to continue to bring this technical expertise into the policymaking discussions. We presented in the white paper 5 open access policy options for DSI benefit sharing, ranging from a micro levy, a membership fee, cloud-based fees, commons licenses, and a blockchain metadata system. I'm not going to have time to get into any of these, But what I was often asked in the talks in the months after we presented the white paper was, okay, so which DSI option of those 5 is the best? And what we started to do in the winter of last year was to really develop data sets on understanding what DSI provision and use patterns were to be able to pick which one would make the most sense. So what we were able to do is take 18 million sequences where the country of origin was known and cited in about 118,000 open access publications. So here's the network diagram for DSI provision from Argentina. So DSI that was sourced from the physical genetic resource was taken from Argentina and then later sequenced. For 84 countries, use data that is from Argentina, where Argentina could claim sovereign rights over it. That's a lot. That's maybe almost half the countries in the world use Argentinian data, or scientists use the Argentinian data. But in a similar way, scientists from Argentina are using DSI from 89 countries. And again, like in the previous slide, you do see scientists from Argentina using slides from neighbors here, Colombia, but also using DSI from much farther away countries like Norway, right, or Jamaica. And this really reflects the integrative type of work that scientists do. We use the data that we need to test our hypothesis. We don't usually seek out data for specific geographical regions. And so our work is highly interconnected with each other. And it's important for policymakers to realize that there are not only trying to protect and to get benefit sharing from the countries that are using Argentinian DSI, but also that they should be able or looking to support Argentinian researchers working in Argentina that are using data from outside of Argentina. That really a DSI policy mechanism needs to reflect both sides of that coin, both the giving and the taking. In fact, Argentina is not unique. So we graphed the data of use and provision. So use on the x-axis, the number of countries that are using, and then provision on the y-axis. You can see Argentina has a roughly equal amount of use and provision. And in fact, the trend line is about the same for all countries, regardless of their economic grouping. G77 being, developing economies, OECD countries being high-income countries, and then BRICS countries being sort of middle-income countries. We see the trend lines being roughly the same. In fact, this is a really important take-home message from the studies that we've been conducting. If you look at the total number of publications done by users in these countries, you can see that DSI tends actually to be used more by locals than by foreigners. What do I mean by that? If you look at the biggest box in each of the bar charts, it matches the country of origin for that DSI. So for example, over here on the OECD, the biggest amount of users using OECD sourced DSI, so high income countries, actually is OECD users. If you look at G77 countries, so low income countries, you see that the biggest amount of users is here, G77 users. If you look at BRICS, Brazil, Russia, India, China, South Africa, again, the biggest block of users is from the BRICS countries. And this means that far more often than some user in a rich country in the global north taking data from a poor country in the global south, it tends to be much more common that actually we use data from nearby, that we have local access to, that we're interested in solving a local or a domestic problem. And ultimately, there's the arrows to illustrate my point. It is also important to realize that the OECD DSI is still used, all of this is used 3 times more than BRICS or G77 DSI. And I would propose, and colleagues are proposing, far more important than trying to lock up the DSI to gain benefits from it is we want to get these bars to rise up. We want BRICS sourced DSI and G77 sourced DSI to be used more, to be able to get more benefits out of them for conservation and sustainable use and for many other purposes. But if we lock up this DSI in a bilateral mechanism, for example, then the ability to benefit from this, to reuse it, will not grow. Finally, it is really important to note that when it comes to talking about capacity building, we still see by looking at the total number of publications that there are 40% fewer DSI-based publications in either BRICS or G77 countries, that there are fewer researchers there and there is a lower total scientific output. And this is the kind of shortcoming or uneven playing field that in the context of a UN negotiation would be important to try to address. Finally, we are talking as members of a newly created network called the DSI Scientific Network about a decoupled multilateral framework for DSI benefit sharing, in which we call for a multilateral mechanism because we believe that a bilateral or a mechanism that requires tracking and tracing will actually disincentivize the use of LMIC or low and middle income country sourced DSI. So if I create conditions on some sequences, but not all sequences, then I create an economic marketplace where I preferentially use those sequences where there are no restrictions. So I grow the bar graph where the OECD bar graph higher, and I don't grow the G77 and the BRICS bar graph. If I want to grow that bar graph, I need to decouple access from benefit sharing to not create a competitive marketplace where some sequences are less attractive to users than others. In fact, what I actually want to do is to create a virtuous cycle. I want to recognize and incentivize the contribution of biodiversity DSI into the databases. And what I really want to do is create this monetary benefit sharing mechanism decoupled from the databases, where the funds actually do get incentivized by more DSI from low and middle income countries coming into the database and creating, for example, a bonus payment where the more that that country has contributed to the open databases, the more they get out of a multilateral fund. But where I have not created a tracking and tracing mechanism that penalizes and restricts the usage of certain kinds of sequences. Finally, if you want to look at the graph that I just presented in the context of the current CBD negotiations, it means that in this diagram, which has been produced by the CBD Secretariat in Montreal, we are really looking for a safe landing space for science that's somewhere around in what's being called options 3.2 or 4, where we really can decouple access from benefit sharing. I wanted to just put a small plug out there. We have, as I mentioned earlier, a new network called the DSI Scientific Network. We are welcoming new members and also offering briefings to different countries. If you're interested, please check out the website here below. And finally, I'd like to acknowledge the co-authors and the many very smart people that worked on this data set to create all of these diagrams and charts, and colleagues at IBK Gottesleben and at EMBL EBI in the UK. Thank you, and I apologize for going a bit late, Fernanda.
Okay, Amber, thanks for such a very interesting presentation. Okay. Remember, the attendees, remember that you can write your questions in the questions and answer chat. Well, our next speaker is Dr. Jane Anderson. Jane is Associate Professor at New York University and has a PhD in law. And her work is focused on the philosophical and practical problems of intellectual property law and the protection of traditional knowledge. Jane is a co-founder of Local Context, which delivers the traditional knowledge and biocultural label system. These digital mechanisms support provenance and contextual metadata for research derived from indigenous peoples, lands, and water. The title of her talk is Mechanisms and Training to Support Researchers. Jane, your time.
Great, thank you so much. Can you hear me okay? Good, okay, I'm going to share my screen. Okay.
Okay.
Great. Well, thank you so much for the invitation to be here, and it's wonderful to follow Amber. I learn so much from her talks every time I hear them, and to be here with Casey as well. I'm here in Lenape-Hoking, the lands of the Lenape nations. I just want to pay my respects to the ancestors and to the present and emerging leaders of these lands and waters. And I'm going to do something just a little bit different to take us in a different kind of direction, thinking about traditional knowledge and the different kinds of questions that arise for Indigenous people and local communities within this kind of larger context of the Nagoya Protocol, access and benefit sharing, and DSI. Interesting. So I guess we're kind of in this particular moment with really big projects focused on genetics and genetic resources, where the significance of bioinformatics and sequencing has become critical to the possibilities of research. And what's important to think about are the different kinds of stakeholders that are part of this conversation, and the different kinds of knowledges and worldviews that are also important to that research, and how to include that and make it visible or keep it connected to the data. And that's kind of key to the talk I'm going to give today. So another thing that emerges in this context is the push towards open science and open data. And this movement towards the open sharing of data is relatively recent. And part of the future of science and thinking about what does it mean to have relationships with particular kinds of communities, and the different ways in which that affects how that data travels over time, and what that might mean to for people who are using that data who don't have those relationships with those communities. And so how can those communities, for instance, know what is going on, or when some value is created, and how can that be shared back? So this kind of question around benefit sharing and kind of making that differently possible. Another thing that we're kind of seeing or start to emerge as well is a kind of focus on the value that Indigenous communities might bring to an area of genetics, particularly thinking that there have been a number of papers that are coming out that are written by Indigenous scholars that really highlight the need for the inclusion of Indigenous voices as a necessary part of research, and particularly reflecting upon the need of those voices within kind of genetic diversity. And Indigenous authors are really part of thinking about what kind of better research looks like, and how to enhance that research environment that they, as different kinds of interested parties. So some of the challenges that are created by DSI in relationship to traditional knowledge and associated traditional knowledge within the Nagoya Protocol are that most national ABS laws don't apply to DSI, which is kind of something that we were talking about earlier. And that embodied traditional knowledge exists within most DSI, but databases don't have provisions or technical capacity, including particular fields for recording associated traditional knowledge. And this means that associated traditional knowledge is largely invisible, and therefore they're kind of really limited ABS options in relationship to that. So the key issues for Indigenous people in this kind of area is how to recognise Indigenous rights and interests in raw data and digital and DSI within an open data environment, how to share DSI and collaborate around ethical use and access in ways that are consistent with existing community protocols, and then how to negotiate equitable outcomes from the use of DSI, including potential for commercialisation. So where this work sits, and it's important to kind of say this work sits within a kind of a framework around Indigenous data sovereignty. And Indigenous data sovereignty is really focused on two elements. It's a discourse around rights and interests, but it's really about data for governance and governance of data. So data for governance being gaining access to use data to transform the lives of Indigenous people, as well as managing access to data to ensure that use is relevant and responsive to Indigenous needs. One of the overriding kind of policy frameworks that has been recently developed are the CARE principles, and the CARE principles are around collective benefit, authority to control, responsibility and ethics, and they're seen as a complementary to the FAIR principles, particularly focusing on the relationship dimension of data, not just the data-centric component, but that the FAIR principles focus on. So to be fair and care is bringing in Indigenous relations into kind of management and circulation of data. And I think what we've seen and what's coming out of these kinds of questions around Indigenous genomic data is the kind of these key dimensions for how to conduct research and what these kind of expectations are from Indigenous standpoints around building trust, enhancing accountability and improving equity. So I just want to talk you through some of the kind of mechanisms and training and guidelines that are out there. Some of these I'll put into the chat when we're finished so you can kind of have access to them. This is the Nagoya Protocol Learning Portal. This is a really great place to learn about the Nagoya Protocol and kind of understand what the implications are for you and your research, and to go deeper into kind of some of the questions that you'll need to kind of focus on. This is the Nagoya Protocol Hub that Amber mentioned before. This is also a really important kind of site for thinking through kind of what are the obligations and responsibilities that you have as a researcher in relationship to Nagoya. This is another site that starts to talk about Indigenous people and in digital sequence information, the different kinds of questions around the resources that are available for thinking through the questions that Indigenous people are kind of thinking through in relationship to DSI. And then these are some genomic guidelines that are coming out of New Zealand, particularly for doing research with Maori communities, and also doing research on particular kind of Tonga species or species that are special to and sacred to Indigenous communities in New Zealand. And these are some of the guidelines that are coming out in relationship to doing research in Australia. Again, particularly supporting the different kinds of ways of building trust and relationship building that are required when working with Indigenous communities. So I want to focus a little bit on a practical mechanism that my colleague Maui Hudson and I have developed in terms of thinking about the different kinds of questions of provenance around data and kind of what that starts to potentially be or could be into the future. These are the traditional knowledge labels that we developed over the last 10 years, working with Indigenous communities to bring digital protocols into the management of data. This has been a very long project, and we're just getting to another dimension of that, which is around the biocultural labels, particularly the way in which data and the kind of enhancing contextual metadata, which is really what these labels focus on, and kind of building in the capacity for people to know where data comes from as it travels across the different kinds of database contexts. The biocultural labels in particular focus on provenance, but also the different ways of thinking about consent and the interests that exist within that data at that kind of moment of conducting research. What's interesting or important is the way in which that doesn't necessarily track across with the future uses of data. So we're trying to imagine a system where those rights and interests are incorporated as contextual metadata that can kind of move across, so secondary, third, fourth, fifth users of that data have some information that can help guide their use of that data. So really what the traditional knowledge and the biocultural labels communicate are provenance, where certain things come from, the different kinds of protocols that might exist in relationship to how that material can be used into the future, and the different kinds of permissions that sit within that kind of moment of generating the data. This is just an example of both the traditional knowledge labels and the biocultural labels being utilised within a context in New Zealand. This is Maui Hudson's community, the Whakatohea Nation. They're involved in a project called the Moana Project, looking at ocean heat waves and modelling currents. And they're doing it kind of around the whole coast that is the region of that community. And on the left-hand side, you'll kind of see the Maruhia Atu, which is a chant, which is a particular kind of song that is sung around the coastline, and the particular places that are significant. And what they're interested in is the overlay between the scientific knowledge and the cultural knowledge, and how the kind of labels then can start to demarcate the kind of relationships between the cultural information and the scientific information. So then on the right-hand side, they're using the biocultural labels as kind of part of the way in which they can indicate their interests in that data as it's kind of being developed, and then it's being shared within this kind of open science contexts. This is just an example of some of what's important about the labels is they're both machine readable and human readable. The kind of icon that you see is the human readability part of it, and the ways in which we've developed the labels allow for the kind of machine readability of the labels. And then kind of thinking about what the ways in which the labels support access and benefit sharing in certain kinds of ways, particularly around consent, whether that's there, whether it's present, the different kinds of research uses that communities are interested in being part of, establishing relationships, and making sure that it's clear where certain kinds of data has come from as it kind of moves into these other spaces. So one of the mechanisms that we've developed and we particularly wanted to bring to you today are the notices. So the labels are one tool, the notices are a second tool, and the notices are really for researchers to disclose Indigenous rights and interests at the kind of point of research. This is the biocultural notice. It's a visible digital identifier, which is really applied by researchers or by institutions in repositories to recognise the rights of Indigenous people and to kind of define the certain kinds of uses of that data that's being generated from Indigenous lands and waters. This is the traditional knowledge notice, which is also similarly functioning as a visible digital identifier, which can be applied by researchers also recognising that there could be accompanying cultural rights and protocols and responsibilities that need to have further attention. This is an example of one of our notices being utilised within a cultural heritage context. This is with particular kinds of photographs. This is at Simon Fraser University. And this is one of the notices being used within Geome, which is a geological and genomic database. This is the traditional knowledge notice being, when the samples are uploaded into Geome, the researcher can indicate if there are traditional knowledge interests and make that disclosure clear when uploading their samples into the database. This is the example of the biocultural notice being used in a Vaccinium database. This is a research that was conducted around a bilberry, which indicates that there are Indigenous interests that need to be kind of considered in the future use around that kind of data. And then this is kind of one of the more recent kind of pieces that we've been working on is kind of how to disclose those interests within publications, and researchers who are really interested in thinking through data ethics and data accessibility, and also thinking around what the disclosure of Indigenous rights and interests might start to look like within these contexts. And this is kind of just molecular ecology, but these conversations are also happening with nature and also with ORCID, particularly because it kind of increases researcher transparency in terms of thinking through what the kind of ethical and equitable frameworks for doing research in collaboration with Indigenous communities can look like. As I said before, the labels and the notices are human readable and machine readable. And this is kind of partly, connecting particular kinds of permanent identifiers with each other. So from ORCID to RAWs to DOIs, and kind of the ways in which those kinds of connections are made, which allow for the kind of traveling across with data across multiple kind of platforms. And then finally, this is just the local context hub, which is where the notices and the labels are generated. This is on soft release in the end of the month for people to engage with and to kind of start thinking through how they might be able to add particular kinds of notices to their data. So I'm going to finish there, and thank you again for your time, and excited to have a conversation at the end of these presentations.
Thank you very much, Shane. Very interesting also your presentation. Well, our next speaker is Professor Casey Bansal. Okay. Professor Bansal obtained his doctoral degree with gold medal from the Indian Agricultural Research Institute. He pursued advanced research from Harvard University, Cambridge, USA, and since his return, he occupied eminent positions as Professor of Plant Biotechnology at the Indian Agricultural Research Institute, and he was coordinator of the National Project on Transgenics in Crops and director of the National Bureau of Plant Genetic Resources in New Delhi. He was the first to be selected for the prestigious Norman Borlaug Chair for Crop Improvement by ICAR in India. Currently, Professor Bansal is serving as Secretary of the National Academy of Agricultural Science and as member of the Board of Directors of the Global Plant Council. He has contributed significantly in the area of plant biotechnology and plant genetic resources. He has published over 150 research articles in international journals, and he has supervised over 20 doctoral students and imparted training to over 500 scientists and scholars at the national and international level. He is fellow of the National Academy of Agricultural Science and of the National Academy of Science in India. The title of his talk is Potential Conflicts Related to DSI. Casey?
Yes, I'm trying to share my slides.
Okay. We can see them.
Yeah, thank you very much. Thanks to the organizers at the Argentinian Conference on Plant Physiology, and also, of course, thanks to Global Plant Council for organizing this particular conference. And very happy to also listen to two very important and wonderful talks by both Amber and Jane. And I'm a plant physiologist myself, so I'm very happy to participate in this international conference on plant physiology, and my best wishes to all the plant physiologists, those who are attending this particular conference. Well, the topic assigned to me, as you can see, is the potential conflicts related to DSI. But my presentation, I will have two different parts of my presentation. One, I'm going to give you an actually speaking overview. I'm a plant physiologist, so kind of finding value with these particularly plant genetic resources for utilization for food security. And then in the later half, I'm going to talk about the issues and the concerns related to the DSI. In fact, quite a lot has already been covered, I would say, nicely by both Amber, mostly by Amber on DSI, but I'm going to give you some kind of conflicting issues or challenges or concerns, which are raised by, or divergent issues raised by the contracting parties to the Convention on Biological Diversity. To start with, of course, I thought there are several students and young researchers and scholars participating in this conference, thought to give them little basic background about, I'm not able to move my slide. Yes, about the importance of plant genetic resources. I'm sure that you all know, but at the same time, it's important to really remember that how important these plant genetic resources are, because they are the genetic material of plant origin of actual and potential value for food and agriculture. And they provide the basic material for crop improvement. They have got genes for resistance to tolerance to various abiotic, biotic stresses, even climate resilience, or for nutrition, and also help in diversification of the cropping system, farming systems around the world. And of course, they do have potential new crops hidden in many of those, plant genetic resources which have not been, mainstreamed in agricultural production so far. And plant breeders require the access, although, on a very regular basis, on a wide pool of these genetic resources to create and develop new novel plant varieties. And countries, this is an important point that we always should remember, that the countries are interdependent for the food production on these plant genetic resources. And due to the climate change and population growth pressures on food and production system, I think it is important that we enhance the use of these plant genetic resources more and more, in different forms. And just to give an example about the use of these plant genetic resources way back in early 60s by Norman Borlaug, brought a revolution on green revolution, we call it wheat revolution, by developing high yielding varieties of wheat, by using these all sort of land races or farmers varieties or obsolete varieties in general plant genetic resources for developing high yielding varieties. So it is so very important. And actually speaking, by utilization of these plant genetic resources by Norman Borlaug in case of wheat, has recognized the importance of not only of science, but also the role of farmers and mostly the role of technology, and that's the conventional plant breeding here, I'm talking about it. And also the public policy. The policy inputs are so very important. Here I'm talking, of course, of the Indian government, and when we had the beginning of this green revolution in 1968, and we were able to harness the benefits of these conventional breeding and the use of plant genetic resources to increase our production rate. Let's say from 1951 to in 1968, you can see it's more than doubled. So therefore, it is important to recognize not only the science behind it, but also the policy issues, which are so very important. I think that's what we are talking about today in this particular panel discussion. But that was possible because of the free exchange of the germplasm that time in 60s or before 1993. But now in 1993, we have a regulated access of plant genetic resources, in general of genetic resources, including plant genetic resources. And CBD, which came into being in 1993, has three different objectives. We all know it's conservation of biological diversity, sustainable use of its components, and fair and equitable sharing of benefits arising out of the utilization of these plant or genetic resources in general. And it provides a sovereignty on to different nations or a country on the genetic resources present in its borders, and also the access, which is not free anymore, which was before 1993, and therefore we had a green revolution. I give an example of wheat, but now if you want to access genetic resources, it has to be, as also mentioned by Amber in different ways, that it has to be based on mutually agreed terms, or with prior informed consent on a bilateral basis. That's what CBD defines, the exchange of germplasm in a manner like this. And then there are, of course, different uses in general of genetic resources, a kind of slide which was already shown in a different way by Amber. So here there are different types of genetic resources, plant, animal, microbial genetic resources. I'm talking with regard to the present CBD in scenario. I've not included therefore here at the moment anything to relate with RNA or DNA or protein sequences. But then mostly, of course, fungi can be added, plant, animal, microbial resources. There are different types of genetic resources. They are then subjected to different uses, either for research and development, again mentioned by Amber, for both commercial and non-commercial purposes, and which is again when we talk of CBD based on prior informed consent or on MAT, that is mutually agreed terms. And it is these genetic resources across different species are used by different sectors, like in biotechnology or in pharmaceuticals, seed and crop production, and many other areas like botanicals and horticulture, or even farm animal breeding. So there is an important, these genetic resources are importantly utilized, for the welfare of mankind at large, I would say, on this planet Earth. And when we talk of conservation, which is the first objective of CBD, we have plenty of these plant genetic resources, particularly I'm now focusing henceforth, we have plenty of these genetic resources conserved globally. We have about 7.4 million accessions in about 1,750 gene banks across the world. Of course, most of them present in many of those international gene banks, which is with CG centers. Like we have ICRISAT in India, we have got IRRI, also in Philippines, and we have got CIMIT, for maize and wheat in Mexico, C8 or SIP for potato or tuber crops in Lima, Peru. So we have a large amount of the accessions of various plant genetic resources conserved. And of course, we have one international gene bank here, which is Svalbard, just to highlight the importance, for posterity, they've been conserved there under permafrost conditions. But with regard to utilization of it, I'm just talking in general. Before 1993, before CBD, and even after now 1993, what is the situation with regard to utilization of these plant genetic resources for crop improvement in general? And it gives you an example here from International Rice Research Institute, which is on rice. If there are about, let's say, more than 100,000 accessions conserved in that IRRI gene bank, only 2% of them you find the percentage of that, 2% you find the ancestry in the commercially released cultivars, which is very less. Those, of course, 11% which has been utilized in crosses. But finally, in the varieties of rice which are released globally, you find that only 2% are there from, some of the large number of accessions which are conserved, in the IRRI gene bank. And of course, the reason is characterization. We have not been able to find value in a particular genetic resource, that which has got for resistance to diseases or to abiotic stresses or for nutrition. So I'm just showing you as a scientist, I'm a plant physiologist, I mentioned, who worked on extensively on genetic resources. So I thought some of the scientists and student friends may like to know about it, that the status of conservation is important, but the utilization and preparing those germplasm for utilization is rather important. Here we're talking about all the germplasm of wheat conserved in Indian National Gene Bank. We have about 22,000 accessions, which we, for the first time at a single site, characterized them all for various characteristics. For example, of course, we developed a core, and then we found new sources of rust and spot blot resistance, and we found new sources, for instance, to powdery mildew, just to highlight the importance. And these are new sources which were not discovered until, let's say, 2020. These papers were published, though the work was done in the last decade or so. And also, we must remind ourselves that we have to deliver these sustainable development goals as a part of the strategy of United Nations, of FAO by 2030. And one of the goals always remain to be reached is end hunger, achieve food security, improve nutrition, and promote sustainable agriculture. So I think we need to give more emphasis on the sustainability, and therefore utilization of these plant genetic resources are really important. But then, as we have been talking about, what the agenda of today's panel discussion is also, of course, that there's a regulated access. We all know about plant genetic resources through conventional breeding. If the products are developed and they are commercialized, they are subject to benefit sharing as a result of CBD, now Nagoya Protocol, very nicely already described by Amber. And also there's a plant treaty, which is having a similar concept or objective as we have got CBD. I'll just talk to you about in a minute. And then these are subjected, the commercial products subjected to benefit sharing entirely, of course, which is important as incentives for biodiversity conservation and sustainable utilization. So not only it gives you a commercial product for food security in case of plants, we're talking about it, but also the benefits to go back to the provider countries or to custodians as incentives for biodiversity, conservation, which is already in practice on a bilateral basis. Of course, in case of plant treaty, I'll just talk to you about in a minute, which is on a multilateral basis. But then when we talk of DSI, this is now the question is that there's a controversy which has been raised by several contracting parties or otherwise discussion going on, that whether DSI should also be subjected to kind of regulated access as we have been observing in case of genetic resources through commercial product development. But then there are certain issues, of course, which we need to define. First of all, the definition itself of the digital sequence information, whether it is RNA, DNA, or as Amber mentioned about some biochemical, components, including protein sequences, etc. And the owner of DSI, is also important to determine that. And as we talk about custodian farmers, those who have been conserving these plant genetic resources for a long time, this is my personal view as a scientist. I think it's important also similarly to recognize the role of researchers and technology developers who have been able to convert these genetic resources into this DSI. And then this has been now all data stored into these, international databases, which are playing a very important key role. In fact, really speaking, I think it's one of the biggest examples we have in the country of multilateralism, also providing a free and open access to the whole public, whether commercial, non-commercial. I think, this need to be recognized. So what is the role of these open access databases is also equally important. But when we go back to now, to the genetic resources, and apart from CBD, which of course we've talked about Nagoya Protocol, now what is the situation with regard to plant treaty? I thought I should give you a little bit an overview of that as well, what's happening on ground at the moment. This international treaty, which also came into being after about 7 to 10 years of negotiations after the CBD came into being in 1993, and with an objective again of conservation and sustainable use of plant resources, primarily for food and agriculture, because genetic resources in general already covered by the CBD, and the fair and equitable sharing of the benefits arising out of the use of these plant-genetic resources for food and agriculture in harmony with CBD and for sustainable agriculture and food security. Here, there's a multilateral system which allows for facilitated access of PGRs for research, breeding, and training for food and agriculture, and recipients of these material, they sign a standard material transfer agreement that specifies the conditions for use and benefit sharing. And the material to this MLS will come from the different contracting parties who are signatory to this international treaty. And also it could come from private sector, or also it could come from other sources which are there in a particular country or a nation. Now, I think there are issues as well. When we talk of benefit sharing, using these plant-genetic resources, I have not come to DSI for that matter yet, what are the issues, but even under the treaty, we have these plant-genetic resources through multilateral system which are being, accessed and shared. And what is the status? There was some kind of expectations which have not been really realized fully yet when we talk of benefit sharing. And also the reason could be that the time needed for the product development through conventional plant breeding is quite long. It could be 10 years, could be 20 years. And if you're using a wild species as a plant genetic resource, particularly let's say for climate resilience, it could even take 30 to 40 years. So only then when the product is developed and is commercialized that you can realize its benefits. So there are issues there as well. And how to access the value of genetic resources? It's a long-standing question. And similarly, I think it's also remained true for DSI as well. And then the multilateral functioning needs to be enhanced, because many of the contracting parties have always been requested in the GB meetings that they should put more of their material under MLS. And if you look into that, I don't have those data actually speaking. I thought I should have it here. Not very many accessions out of what is being available. I give you a figure, globally. Only I think very little of it has been put under this MLS. For example, from India, which I know, of course, I was instrumental there, that we have out of about 4.5 lakh accessions, or I should say 0.45 million accessions, only 26,000 accessions of 9 crops have been put under MLS. So similarly, Germany, I believe, is the only country where 100% has been put under MLS. So therefore, there are many other countries who have not been able to put and realize this kind of functioning of MLS. And sharing of commercial benefits also need to be enhanced. And that's the reason that their expectations of benefit sharing have not been yet fully realized. So when we talk of DSI, and what are the issues, while there's a convergence among the different contracting parties to CBD, that yes, DSI, of course, are used to support conservation for sustainable use and including taxonomy, breeding, public health, and food security. Amber also mentioned about it. But then, however, there are several divergent views regarding the benefit sharing from the use of DSI on genetic resources. Here, for example, already there is a concern that the expected benefits are not shared with the contracting parties, and there are very many difficulties, as Amber explained it very well. Then DSI will circumvent, this is another fear that contracting parties have, some of them, the DSI will circumvent the physical access, of the whole genetic material, and the use of genetic resources therefore will avoid or reduce benefit sharing obligations. And then on the other hand, of course, there are contracting parties which believe, that the regulation should remain open access, and if it is regulated, DSI, if it's regulated, it will affect innovation. So we have got both types of views, and therefore these are some of the concerns. And there are other challenges, related to the use of DSI. For example, many contracting parties are questioning the fact that whether DSI by itself falls within the scope of the ABS. Because what we have talked about Nagoya Protocol so far, not DSI, but the physical access to whole genetic resources. And there are difficulties, I mentioned, not only with PGR, also difficulties in determining the value of DSI. For example, what could be the value of a single sequence used in commercial products? Identifying providers and users. And though, again, a lot of emphasis given and clarity was there, from members' presentation on this, then determining provenance of material and also traditional knowledge tags, monitoring of commercialization of these products and traceability concerns of DSI use. So there are several of these concerns which have been raised by different parties. But then also, actually speaking, this is at times quite disturbing when you read about all this, that many developing countries have even indicated that they will not support the adoption of post-2020 global biodiversity framework. Now, of course, it is in 2022 is going to happen, this meeting, that does not contain a benefit sharing solution for DSI. So there are, really speaking, divergent issues about it, and we need to find some certain mechanisms. And if you start comparing and look at the bilateral versus multilateral approach, though CBD is always bilateral, and it says that specific benefit sharing conditions related to DSI resulting from utilization of genetic resources could be included in mutually agreed terms, as we talked about it before, which is the case with the whole genetic resources. Or there could be a multilateral approach for DSI, which will provide an alternative to requirements for prior informed consent and mutually agreed terms, and therefore help to reduce transactions costs and facilitate equitable sharing of the benefits. Mechanisms needs to be developed. Again, Amber gave several options, and I believe multilateral approach is already, that's been used, in fact, by sharing this data through public databases to many of those users from the international nuclear sequence database collaboration. I think that's already serving the purpose of a multilateral approach. But important is that, again, as scientists, that we should try to increase the capacity to access, use, and generate these and analyze digital sequence information on genetic resources, because only 2% of the global biodiversity has been sequenced so far. So rather than regulating, my opinion as a scientist remains that it should be facilitated, the use of DSI, or even generation of use and analysis of DSI by even those user countries or provider countries who don't have that kind of a capacity. I think they need to enhance that capacity to generate this data and utilize this data using some of the modern tools like genome editing. So therefore, at the Global Plant Council, we have a discussion, and we believe that plant scientists also need to be listened carefully in these policy matters, both developed, I would say, nationally or internationally. I think so far it's only most of the bureaucrats or the lawyers, or others who have been attending these governing body meetings or various, whether CBD you talk about it, I have the experience of international treaty. I think the scientists should have a voice, because these are the scientists who are using whether whole germplasm resources we're talking about it or DSI. So therefore, our message is that DSI to remain open access to support science and innovation, not only support science and innovation, but also protecting the biodiversity and sustainable utilization of it, which are the real objectives of whether you talk of treaty or the CBD. So, and a balanced approach is required both for monetary and non-monetary benefits. And then monetary benefits are very important for some of the developing countries through technology transfer or partnership and collaboration, information exchange, and capacity building to prepare them to utilize their own resources, which are indigenously developed, for producing novel varieties for climate resilience and for other, food-related, traits. So therefore, unlimited and open access of DSI is needed for sustainable use of biological diversity, for meeting the challenges of climate change, food security, and sustainable development goal I mentioned to you about it. We should have a reminder by 2030. So I think as scientists, we feel that we should facilitate the access. We must utilize those germplasm resources, whether whole germplasm resources as seeds or DSI. And there should be, of course, policy, discussions which are going on, internationally, and only the scientists should be heard so that we can try to really speaking a situation which is win-win kind of a situation, and we don't curtail, the kind of use of these. We rather facilitate more and more, because we need to make a progress for the, increasing production for the burgeoning population, likely to be like 10 billion, by 2050 or so. So we have a long way to go. I think we must learn from our past, experiences. We have experience, I mentioned about ITPGFR itself. It's not that successful as far as I know, though there are now changes taking place, for enhancing the MLS and also benefit sharing. So looking into all that, I believe open access remains as one of the mechanisms. Of course, we need to develop more into it. So with this, I thank you, and thanks again to the friends in Argentina organizing this particular workshop or a conference on plant physiology, and of course, Global Plant Council for giving this opportunity. And thanks, Fernanda, for your nice introduction and supporting this workshop and on this important issue. Thank you very much. Thank you, friends. Thank you, all the listeners, please. Thank you.
Thank you all for your such interesting talks. Well, for me, I when I saw one of the slides of Shane that says Nagoya what? That was my first impression when I first heard about the Nagoya Protocol. So I feel relief when I saw that I am not the only one who say Nagoya what? What is it about? Well, we have some time for questions. Here we have one from Veronica Lia. She says, thank you very much for the great talks. What do you think is the best approach to deal with the use of germoplasm museum collections that were established prior to CBD or Nagoya and for which no PIC is possible? You can read it if I didn't. You can ask, you can read it in the question and answers in case I read it.
Probably KC is the most informed.
Is the most? Okay, yes. Yeah. Okay.
KC,
we cannot hear you.
No, in fact, any collection prior to CBD, as per the national legislations, which are there as a fallout of the CBD, this internationally legally binding instrument, any material which was collected before 1993, which is in your national repository, in fact, these do not apply. But as far as, the material collected by CG Center or conserved in CG Center gene banks, before 1993, whatever was collected and conserved as ex situ collections in these CG Center gene banks, which I mentioned, there are several of them in the world, they have that material in trust available with FAO. And that material, in fact, is utilized, without any bilateralism or without any kind of system which is imposed by CBD. And therefore, we have this international treaty where there's a multilateral system and that goes, an easy access through, of course, a standard material transfer agreement, which every provider and the user, even they go subsequently, they have to utilize, that SMTA, for providing that material. Yeah, I hope I have been able to clarify a little bit about that. So this is the material which is accessible, collected before 1993 and available with country. And of course, the sovereignty will remain with a particular nation, whether they will have that kind, they have the right, of course, whether that they will allow or not, or they will have their own, mutually, agreed terms. On that basis, bilateral that could be provided, But when you talk of international treaty, of course, whatever is material, parties are all supposed to be sharing that material with MLS collected before 1993 as well.
Maybe I could follow up on Casey's things. There are some, this is not true for every country, but so in general, as Casey indicated, great, you're lucky is the answer to the question, I think before 1992. However, for some national laws do require that ABS permits are needed when you take material out of their ex situ collection. So for example, in the microbial space, if there are material that was sampled before 1992 and then lands in a collection and then is taken in 2021 out of a collection, then for example, I'm familiar with the Spanish law. In Spain, you have to ask for a Spanish PIC, as a prior informed consent, to get it from that new collection for that new use. So the access is actually considered access from an ex situ collection. The perverse thing that can happen is if exactly that identical microbial strain had been deposited here at the DSMZ in Germany, those Spanish laws don't apply to the German collection, they only apply to the Spanish collection. Something similar can also happen under French legislation, and I'm not sure about other countries. These are like real, like legal specialty answers, but really funny things can happen just depending upon the national law. So Casey's advice is 100% correct, but then you sometimes also have to look at the individual national legislation on top of that.
Yes, exactly.
Okay, someone else? Don't be shy.
Yeah.
Okay. It says, and I don't know, Antje, it's that right? I don't know how to say the name. It says, as Casey appeared at the end of his talk, plant scientists should have a voice in the policies developed. What ways could you suggest? How scientists could get more involved, especially since most are a bit overwhelmed with all the intricacies of Nagoya and so on.
Yeah.
So anyone else would like to take it up first? Anyway, see, the message is in a way that when we talk of utilization, see the effort or the goals of the conventional CPD or treaty are very clear, you know, not only conservation, utilization is very important. And utilization means by scientists, by utilizing them for various means, for various products. You know, scientists, I mean, of course, from both commercial and non-commercial angles. I'm talking mostly as a public scientist, you know, and for the benefit of the mankind. So I believe while benefit sharing is important, you know, which is the third objective, and those are governed by various rules and regulations, we're talking about it, but sometimes scientists, and I'm sure Jane would really appreciate that scientists may not be able to understand those. I even mentioned by Amber, you know, that these Nagoya protocols should be simplified. So until unless these are simplified, and in fact scientists are made aware of all those intricacies related with benefit sharing or these, you know, legally binding instruments, many of them are not aware, I must tell you. You know, even legislations we have, for example, in our country, many of the scientists are supposed to know it, but then we have to be aware. So what I'm saying is ultimately it is the critical mass of those scientists globally who have been utilizing these germplasm or plant genetic resources, in general genetic resources, either for converting them to DSI or finding these different technologies and means to save time and to save energy and produce more from less. That's the whole idea, to make it more sustainable. Like we do have conventional plant breeding methods available, but some of the traits we cannot, you know, convert them to, you know, the way we can do it using genome editing for that matter. If you want to change only few nucleotides in a promoter sequence, you cannot do that. I mean, of course, you can achieve that by chance, by selecting for a trait through conventional breeding. It will take 20 years. But here you can use 2 to 3 years. So therefore, scientists should be always consulted. I would say this in a way, as scientist myself, practicing biotechnologies for the last 30, 40 years, that, you know, what is the way forward of not only conservation of these genetic resources in different forms, even utilizing them more, because I said before, it's whether plant genetic resources or in general, when you talk of converting them to DSI, 2% only have been utilized so far. And we have those, you know, formidable challenges to talk about related with climate change or malnutrition in different developing world, including India, you know, or you know, and even producing more for the whole population. So therefore, I believe, you know, mechanisms could be different, that they should have more consultations among themselves, more national academies should come forward, you know.
Yeah, exactly.
They should come forward, have a discussion on it, develop a white paper, as has been done by Amber, for example, using DSI, wild DSI, you know. So more and more such efforts should be, you know, converging into a particular common platform, you know, that yes, this is what we feel, you know, and that message should go clearly to the governing body of these different internationally binding instruments, you know, whether it's, yeah.
No, sorry, Casey, I just completely agree. And I just want to back you up on that. I think a couple of practical tips. I put a link in the chat, just by coincidence, following up on the Open-Ended Working Group 3 meeting. There is right now an open call for input from the CPD Secretariat on digital sequence information. You know, you can just write a one-page letter and just say, you know, we use DSI, we use it for, you know, food and agriculture. We're really worried about open access, and we're really worried about compatibility with other, you know, international treaty. You don't have to make it complicated, but I think what's really been missing, and what I've heard colleagues in the CPD Secretariat also say, is the voice of scientists, particularly from low and middle income countries, has not been heard very well. So given that this is an Argentinian, you know, plant physiology meeting, and you have, you know, hosts here in connections with the Global Plant Council, then are there ways that you can write a one-page letter, you know, working with the GPC and with your society to write a half a page and say, like, we're paying attention, we support these principles, we support benefit sharing, but we're really concerned about a loss of open access, and we're concerned that, you know, our voices aren't being heard or we're not being asked. I can also, another practical tip, organizations that scientific organizations that are global in reach tend to be the ones that find a seat at the table more easily. So regional ones mean that the Secretariat has to include multiple to guarantee equal representation. So a body, for example, like CGIAR or Global Plan Council, where they can claim global outreach, tend to get a seat at the table more easily. I work, for example, with the Global Genome Biodiversity Network, and because they're global and they have global representation, they often get a seat at the advisory committees. Same thing is true, for example, also in the microbiology space, the World Federation for Culture Collections, because again, global rather than regional, those are the kinds of scientific organizations that more easily get heard when it comes to expert groups. But then you have to be in a position as a society to be following the CBD website, and when there's a call for nominations, being able to react to that. So there's just there's always kind of, you know, procedures that you have to sort of be aware of too.
Excellent.
I'm also
excellent.
happy to offer, I can put my email there, but if you have, if anybody wants to do outreach, I'm also happy to connect you with people that do that through my network.
I think Isabel is here. We would certainly like to take up your suggestion you mentioned about GPC and you know, and this to prepare a kind of a note, and maybe we would like to have your input as well. I mean, as panelists, this particular, you know, workshop, together we can make this use as an opportunity and I think prepare a note, you know, and send it across.
Yeah. I agree. In fact, we are already working with a statement for the end of this month on climate change, so we can start working on a new statement for DSI, specifically for this call that Amber just shared. I think it's a great opportunity. We will work on that, and I will contact you again, Amber.
Yeah, is absolutely. And like I said, you don't have to reinvent the wheel. You can recycle things. And I know, for example, Michael Halewood with CGIAR is also active in this space, and I'm sure that you could connect and recycle. There's probably some overlap in your positions, I would say.
Perfect.
But did you want to just put, I'm just thinking, do you want to put into the chat the DSI scientific network that you mentioned as well before? Because that also sounds like it would be a great place for people to connect in order to kind of consolidate and think about what that participation can look like and what that contribution can be.
You're exactly right. I should have also said that. so Casey and I are both part of this DSI scientific network that I mentioned also sort of at the end of my talk. And we're also, you know, people that are really, you know, want to sort of get involved in this policy process are welcome to come and work with us. So we you know, work in both directions, to scientists and to policymakers.
That's right.
Okay.
Yeah, at the GPC we are also putting together a resource page in order to help the scientists navigate all the information. So it's for I would say for newbies on Nagoya, so they're really not experts. So it's a good way to go in and afterwards to jump to other web pages and other resource hubs like the one Amber mentioned, because we have a link to them in order to expand on the notion.
Okay.
And if I can also just, I mean, shamelessly also give you my 2 cents. You know, Argentina as a country is very active in the current CBD discussions. You know, there are 200 countries in the world, but they don't all speak at the negotiating table, and Argentina will play an outsized role in this. So to Fernanda and to other Argentinian scientists, you know, go on the ABS clearinghouse and look up the email address of the person that's responsible for this issue on your behalf. You know, these are people that are representing Argentina. And if they're not reflecting what you think about DSI, then now this is the last window of opportunity that you have to influence the process, because their positions are going to be solidified in the next month or two to lead to the in-person negotiations in January. So now is the time to write them a letter, you know, in your local language, saying who you are and saying that you're watching this and that you're concerned.
Yeah, it was a pity we couldn't make, we invited one person from the Ministry of Environment and to Joaquin also, but they couldn't come here because they have another meetings. So it was a pity, but our idea was to bring someone from Argentina to this discussion also. Yes, but we couldn't.
Even if they're not here, you know, an email or offering up, you know, a smaller meeting with just a few people or a discussion. I can also give you the slides that are specifically about Argentina users and providers if you want to share them with them and tell them, you know, what you're thinking.
Yes, we can send them also the link. The meeting is recorded, so they can hear also all the presentations, and we can do that. Go back to Global Plan Council if you want. Okay. Well, anyone else? Some more questions? No? Well, I think we are right on time. Isabel, do you want to add any comments?
Not really comments, just thank everybody for being here. Thank you for the Argentinian Society for inviting them to for this opportunity to organize this panel, and that's all from my side. Thank you so much, Fernanda.
Oh, well, we would like to thank the Global Plan Council again, and particularly Amber, Shane, and Casey Bansal. It was really interesting, your presentations, and I think it will help us to know how to move from now on. The Nagoya Protocol or the DSI is not being applied. We don't know much in Argentina about it. We are not using it, but I think we are going to catch up with the idea in the future. So I think it was a nice introduction. Well, it was a pleasure for me to coordinate this panel also. Remember that during the afternoon we have a new workshop about Meet the Editors. It would be really interesting also for the students. And well, we wait for you in the meeting that officially starts tomorrow. So thank you very much.
Thanks again. Bye.
Okay, thank you.
Thank you. Thank you.
Thank you. Bye.
Thank you, Fernanda. Thank you.
No,
thank you, Amber. See you later. Jane, see you later. Bye, Isabel.
Bye-bye.
Bye.
Okay.
Okay, it is still recording.